Okay, so we've talked about how pigment absorbs light and how that energy is transformed in photosynthesis. Now we'll go into more detail describing the two photosystems that are used to make ATP and NADPH, how they're connected, and how the energy is used to fix carbon in the Calvin cycle. So, studies in the 1950s with algal cells showed that when you combine far red and red light, you get far more photosynthesis than using either light alone, just using either the far red or just the red. You get more than both of these. And they called this the enhancement effect. And this led to a correct hypothesis that there were actually two photosystems. And those are photosystem 2 and photosystem 1. And photosystem 1 was discovered first, but actually 2 starts the pathway. So recall from the last video that light energy is captured by the antenna pigments, and you get an excited electron, and as that electron returns to the ground state, it transfers energy via resonance. And that resonance energy is transferred until it reaches the reaction center. And the reaction center, it transfers its electron to an electron donor. And this is basically what's going to happen in this photosystem 2. So that's the first one we're going to begin with. The process of transferring electrons occurs in both photosystem 2 and in photosystem 1. And as I mentioned, it's called photosystem 1 because it was discovered first. But let's start here at photosystem 2. So the electron acceptor is called pheophytin, and it's reduced with this high energy electron from the reaction center, from those the special pair of chlorophyll molecules in the reaction center. And this electron is then transferred down an electron transport chain that's embedded into the thylakoid membrane. And this works just like the electron transport chain in the mitochondria, in that it's going to produce a proton gradient, and that proton gradient is going to be used to produce a proton motive force that will be used to generate ATP via the ATP synthase. So that's pretty cool, and let's take a deeper look into how that happens. So the first, as we mentioned, the first molecule here that's reduced is pheophytin. Pheophytin then transfers the electron to plastoquinone, or PQ. And when this happens, so PQ is going to shuttle the electron across the thylakoid membrane to the cytochrome complex. And these molecules have different redox potential, just like the electron transport chain in the mitochondria. So here we have the highest redox potential is the cytochrome complex relative to pheophytin. And so this release of potential energy as you transfer electrons from molecules with low redox potential to high is going to be used to pump protons into the thylakoid lumen. Okay, so we're actually going to concentrate the protons in the lumen, right, of those thylakoids. So a little bit different than the electron transport chain in the mitochondria, where we're getting protons in the intermembrane space. But the proton concentration in the thylakoid membrane, it increases 1,000-fold. This is huge. And this electrochemical gradient contributes to the proton motive force. So as these protons flow back down their concentration gradient, this electrical chemical energy is going to be converted into mechanical energy. So this is going to turn the ATPase, and it's going to lead to the phosphorylation of ADP. So recall that in the mitochondria, we called this oxidative phosphorylation. And in the chloroplast, we call it photophosphorylation because we use light. Although the process of chemiosmosis and the proton motive force and the mechanism by which the ATP synthase generates the ATP is all the same, our original energy came from light. So we had one extra energy conversion in this system. So that's pretty cool, right? So we've generated now ATP in photosystem 2, and we're going to use that ATP to make sugar. But we also, as you recall from that first picture, we're also going to generate NADPH. And that's going to happen in photosystem 1. So photosystem 1 has the antenna pigment, and so photons strike them, and the energy transfers via resonance to the reaction center. And the special pair in the reaction center, those electrons are excited. And then those electrons are carried and passed through a series of different proteins. They're eventually passed to ferredoxin, and then to the NADP plus reductase. And what that does is it reduces NAD plus into NADH. So now we've made NADPH. Sorry, I keep forgetting the P. Okay, so we've generated ATP and NADPH. So that's going to be our chemical energy we're going to use to make sugar. That's why the light reaction is important. But recall from the enhancement effect that these systems we know interact in some way. And this led to the development of this model called the Z-scheme that describes how photosystem 1 and photosystem 2 interact. And it explains how the electrons in the special pair of chlorophyll molecules are replaced. So here in photosystem 2, we have the reaction center is called P680, and it's lost its electrons because it's transferred those electrons to pheophytin. And the electrons here are replaced by the splitting of water. So water is going to be split apart, and it's going to transfer electrons to the chlorophyll molecules in the reaction center, and we're going to end up with oxygen gas. So this is how the water becomes oxygen in photosynthesis. So we've replaced our electrons in photosystem 1 by the splitting of water. So recall that we made ATP by using in the process of photophosphorylation, and this ended with this cytochrome complex. So this is holding on to the electron, and that electron is actually going to be the same electron that's going to replace the electron lost in photosystem 1. And it's this special protein or molecule here called plastocyanin, or PC for photosystem connector. And what it does is it carries electrons from the cytochrome complex through the thylakoid membrane and to the P700 reaction center to replace those electrons. So this is the shape, as you can see, of a Z. So if you think about every time the electrons get excited, it raises the energy, and then it comes back down, and you have photons again raising the energy and coming back down. So this makes a Z, and this is how these two photosystems are connected and work together to make the energy required to make sugar. So this is everything, again, all connected, and this is called the non-cyclical flow of electrons. So here we have photons striking and moving through photosystem 2, and PC then connecting photosystem 2 to photosystem 1, and you make NADPH and ATP, and you have this proton gradient. But we also know now of a new system or a new connection where instead of reducing NADP plus in photosystem 1, that excited electron will actually be transferred to ferredoxin, but instead of going to the NADPH reductase, it'll transfer back to PQ, which will then transfer its electron to the cytochrome complex to generate a proton gradient to be used to make ATP. And so this drives some of the photophosphorylation, but not as much of the NADPH production. So these are just slightly different, and we need this one to make sugar in the Calvin cycle, this top one, the non-cyclical flow of electrons. So far we've talked all the way through our light reactions, and this was all happening in the thylakoid membrane and the lumen of the chloroplasts. and we generated ATP and NADPH. And now these high potential energy molecules are going to be floating around in the stroma. And this is where carbon fixation is going to take place. And it's going to take place in the Calvin cycle, named for Melvin Calvin, who did a lot of work figuring out this cycle. Okay, so here's the Calvin cycle. And the Calvin cycle is broken up into 3 stages: fixation, reduction, and regeneration. So let's take a look at this first step called the fixation of carbon dioxide. And in this step, carbon dioxide combines with this compound called ribulose bisphosphate, or RUBP. The 6-carbon compound then splits into a 3-carbon molecule of 3-PGA. So we have 2 3-carbon compounds. And this step of fixing CO2 to ribulose bisphosphate, or RUBP, is mediated by this enzyme called ribulose 1,5-bisphosphate carboxylase oxygenase, otherwise known as Rubisco. And Rubisco is found in all photosynthetic organisms and is used by the Calvin cycle to fix carbon. So this is an enzyme, and it has 6 different active sites in it. And it's thought to be the most abundant enzyme on Earth. So there's a lot in individual plant cells. This enzyme is very concentrated. But it's actually quite slow, and so it takes a really long time to catalyze this reaction between CO2 and ribulose bisphosphate, or RUBP. And in addition to being slow, oxygen can compete with CO2 for these active sites and catalyze a reaction with RUBP. This reaction with oxygen is called photorespiration, and it costs energy, so it's not super advantageous, but it's thought to be used for plant signaling. So let's return to our diagram here of the Calvin cycle, where we finished fixing carbon. The next step that takes place is the reduction of the 3-PGA to G3P, okay? And this G3P is important because it's a precursor to glucose. And because what we're going to be doing in this step is reducing this molecule to this sugar precursor. And so that means we need energy and reducing power. And so this is where we're going to use that ATP and the NADPH that we generated in the light reactions to fuel the reduction to G3P. And some of the G3P is going to be used to make sugar. But some of it is actually going to be used to regenerate RUBP. So the remaining G3P is then used in reactions that use ATP to regenerate RUBP. So that's the third stage, is regeneration. So photosynthesis, of course, like any pathway, needs to be regulated. And so if there's a lot of light and you're making a lot of ATP and NADPH, this would be a good time to make sugar via the Calvin cycle. And so light actually also triggers the production of photosynthesis proteins and activates Rubisco. So these processes are linked. And we also want to shut it off if there's a lot of product around. So high sugar inhibits photosynthetic proteins and stimulates the production of proteins that actually process and store the sugars instead of generating them and making them. As we pointed out before, low CO2 inhibits Rubisco and oxygen binds instead. So these are just pathways, or these are different mechanisms by which photosynthesis can be regulated. And finally, what happens to the sugar that's produced by photosynthesis? It can be used to produce glucose in a process called glucogenesis. And commonly that glucose combines with fructose, and that makes sucrose, which is a disaccharide. And this is water soluble, and it's transported to all parts of the cell and then metabolized in the mitochondria of plant cells via cellular respiration to make energy for the plant. And if energy isn't needed, it's stored, that glucose is made into starch, that polymer, and then it's stored in leaves and roots until it's needed. So that's all that I have for photosynthesis, and I look forward to seeing everyone in class. Thanks, bye.